So welcome back again. This one is water soluble vitamins in the coenzyme part 2, which is done, which is used in continuation of the first part, the introductory part, which is deals about vitamins, coenzyme, cofactors, and the classification of enzyme. Means that water soluble vitamins, especially vitamin B1, B2, B3, B5, and the B6. And now today we try to see the or folic acid coenzymes. So folic acid is reduced within cells, mainly in the liver, where it is stored to tetrahydrofolate, and the enzyme responsible for this is dihydrofolate reductase, and this dihydrofolate reductase can be requires an cofactor known as NADPH. So NADPH and the dihydrofolate reductase can be used for in couple or in combinations as to responsible for this folic acid. This one is a mechanism how this folic acid coenzyme can works with NADPH, and this folate can be converted to dihydrofolate, and the dihydrofolate can be converted to tetrahydrofolate as shown in this mechanism. So this tetrahydrofolate have 6-methylpentane side, and the PABA side, or PABA means paramino benzoate, which have a glutamate substituent on its structure, and this tetrahydrofolate can also be H4 folate, can depict like this. So this one is the structure of tetrahydrofolate. These tetrahydrofolate derivatives, and the function of tetrahydrofolate derivative is to carry and transfer various forms of one carbon unit during biosynthetic reaction. This one carbon unit are either methylene, or formyl, as well as formimino, HNH, and one carbon group can bind to either N5 or N10 of that tetrahydrofolate structure. The most reduced form of tetrahydrofolate carries methyl group, and the most oxidized form carries methynyl, formyl, and formimino groups. This tetrahydrofolate derivative, as I mentioned before, N5-methyl tetrahydrofolate, N5-N10 methylene tetrahydrofolate, N5-N10 methynyl tetrahydrofolate, or N5-formiminol tetrahydrofolate, and so on. The formation of tetrahydrofolate forms. This tetrahydrofolate can be oxidized to N5-N10 methylene tetrahydrofolate, and during this time the serine react with tetrahydrofolate so as to produce N5-N10 methylene tetrahydrofolate plus another enzyme known as glycine. So serine can be described as a principal source of one carbon unit in the reaction. So N5-N10 methylene tetrahydrofolate can also be reduced to N5-methyl tetrahydrofolate, or oxidized to N5-N10 methynyl tetrahydrofolates. So this N5-N10 methynyl tetrahydrofolate can be converted again to N5 formation of formimino of tetrahydrofolate, or N10 of formyl tetrahydrofolates. This N10 formyl tetrahydrofolate can also be synthesized from tetrahydrofolane in the presence of an energy ATP plus another amino acid known as formate. So formate plus tetrahydrofolate plus ATP so as to produce N10 formyl tetrahydrofolate plus ADP plus PI. So N10 formyl tetrahydrofolate can be reversibly isomerized to N5 formyl tetrahydrofolate, and the N5 formyl tetrahydrofolate can form N5-N10 methynyl tetrahydrofolate. These tetrahydrofolate have different function, and this one carbon transfer reaction are required in the metabolism of amino acid, especially serine, methionine, and the glycine, purine, and the thiamine nucleotides of DNA, as well as choline. So these tetrahydrofolate have such function, and this one is the synthesis of methionine, which requires 5-methyl tetrahydrofolate, and this 5-methyl tetrahydrofolate can be converted to tetrahydrofolate, and this tetrahydrofolate again converted to homocysteine, which is in the presence of methionine cyanidase enzyme can be converted to methionine. Methionine means the one that have sulfur CH3 bonds. We can also synthesize glycine from these tetrahydrofolates by using N510 N10 methylene tetrahydrofolate, which donates what 1 carbon unit in synthesis of glycine from carbon dioxide and the ammonium ion. So the reaction seems carbon dioxide plus ammonium ion plus N5N10 methylene tetrahydrofolate in the presence of NADH to produce glycine plus tetrahydrofolate plus NAD. So glycine also made from serine in the react with tetrahydrofolate in the same fashion as above. So this conversion of serine to glycine is the major source of 1 carbon unit, and the serine can be made from phosphoglycerate. Thus 1 carbon unit can be derived from a carbohydrate as a precursors. And also tetrahydrofolate can be also used for synthesis of purine. Purine, which is an isotopic supplement identified origin of carbon atom in a purine ring. Format is in the form of N10 formyl tetrahydrofolate, and this amide in glutamine, the nitrogen, which is in the purine, can be from amide of glutamine, and the format aspartate glycine, and the format in the presence of carbon dioxide can also be used for the component of this purine synthesis using tetrahydrofolate. The same is true with thymine, also synthesized using tetrahydrofolate. This methyl group of thiamine, especially from pyrimidine nucleotide, comes from N5NT methylene tetrahydrofolate. Thus, this uses the enzyme thymidylate cyanidase. N5NT methylene tetrahydrofolate is oxidized to tetrahydrofolate. So this dihydrofolate is then reduced to tetrahydrofolate, and the dihydrofolate reductase enzyme can be used in the catalyst of this reaction. So dihydrofolate plus NADPH to produce tetrahydrofolate. So dihydrofolate can be used for what the production of tetrahydrofolate. This one is also the mechanism. So have a look this diagram which shows what how N5NT methylene folate can be converted to 7,8 dihydrofolate, which in turn form to what folate in the presence of PLP pyridoxal phosphate. Serine can also be converted to glycine. So this one is how the mechanism can be takes place. Methylation of DNA can be takes place by using SAM. You know that SAM is S-adenosyl methionine, which is also always the source of methyl group. Especially in chemistry of natural product chapter 1, we try to depict this scenario of secondary modification, which can be mediated or supported by coenzyme SAM, which is always a source of what methyl group or methylation. In the same fashion here also methylation of DNA can be takes place in the presence of what SAM. So methylation of DNA involves SAM, S-adenosyl homocysteine. The major target of methylase is at the fifth position of cytosine of a DNA. The degree of methylation correlates with transcriptional activity. So this comes from N5-methyl tetrahydrofuran, which one is a methyl group or CH3 group. As the other vitamins, this vitamin or folic acid also found in diet. So many food we consume in day-to-day can have this folic acid. Especially this folic acid is obtained primarily from yeast, leafy vegetables, foliage, hence named folic acid, and animal liver. Animal cannot synthesize PABA, para-aminobenzoic acid, nor attach glutamate residue to nucleic acid. So they require the folate in the diet. So folic acid is susceptible to heat and oxidation as well as ultraviolet light. Therefore, some things that are folic acid or source of folic acid can be highly susceptible for such phenomena or for such thing. So it needs care. It needs cares. The folate deficiency 1 results in complication nearly identical to those of described for vitamin B12 deficiency. The main effect of folate deficiency on cellular processes on DNA synthesis, especially this is due to an impairment of DTMP synthesis, which lead to the cell cycle arrest, especially in S phase of cell cycle rapidly proliferating cells, in particular hematopoietic cells. So the result is megaloblastic anemia as for vitamin B12 deficiency. So The inability to synthesize DNA during erythrocyte maturation leads to what? Abnormally large erythrocyte termed as macrocytic anemia, because what? The deficiency in DNA synthesis. Folate deficiency may be induced during what? Cancer therapy also. So it is very crucial to have what? An optimum folate or folic acid in our meal. The folate acid deficiency or folate deficiency 2 mean that folate deficiencies are rare. This folate deficiency is actually rare due to what? Adequate presence of folate in a food. Poor diet, example, chronic alcoholics can lead to the folate deficiency. The main cause of folate deficiency is non-alcoholics are impaired absorption or metabolism, or it can be increased demand for the vitamins. So the predominant condition requiring an increase in the daily intake of folate, especially for pregnant women, this is due to the increased need for cell division and DNA synthesis. So the need for folate will nearly double by the third trimesters of the pregnancy women. If that's maternal deficiency of the folate can cause a neural tube defect in the fetus, including spina bifida, which is associated with the risk of spinal cord. This one is the diagram of spina bifida, which is associated with the deficiency of folate in pregnant women. So certain drugs such as anticonvulsants and oral contraceptive can impair the absorption of folate. These anticonvulsants also increase the rate of folate metabolism as well. So this folate deficiency may also associated with an adverse effect of different drugs in our body. Different analog of tetrahydrofolates for therapeutic purpose are there. For example, methotrexate, a structural analog of dihydrofolate, is a potent competitive inhibitor of dihydrofolate reductase and used for chemotherapy for cancer. So it acts as by blocking TMP synthesis and appeals proliferating cells even if not malignant or side effect. And trimethoprim is a folate analog which is effective inhibitor for bacteria and the protozoan enzyme, but has a minimal inhibition against the mammalian enzyme, so used as antimicrobial to treat infections. So the next enzyme is cobalamin, which is called vitamin B12. This vitamin B12 includes the free vitamin and the two active coenzyme. These two active coenzyme is known as methylcobalamin or 5-dehydroxyadenosylcobalamin. So vitamin B12 is composed of the complex tetrapyrrole ring structure called core ring, which have a cobalt as a center, and which is similar to that of the porphyrin of hemoglobin, which have iron 2 plus at the center. Therefore, cobalamin means because of having a core ring, having a cobalt at the center. So have a look at this structure. In the structure, you see the red one, which is cobalt plus 1, which acts as a porphyrin for the vitamin B12. Actually, the structure of vitamin B12 is somewhat bulky, but in this core ring system, it has cobalt plus 1. This is the methylcobalamin and then the 5-dioxyadenosylcobalamin. 5-dioxy is at the 5 position to have deoxy sugar as the structure shown, and methylcobalamin means that the methyl group or CH3 is directly bonded with cobalt. When you see the absorption of vitamin B12 for cobalamin, this vitamin binds to the protein originating in the salivary gland and is swallowed along with meat. So the vitamin must be hydrolyzed from protein in order to be active, and the hydrolysis occurs in the stomach, supported by HCl and the pepsin for the intestine by trypsin digestion, as well as releasing the vitamin. So the vitamin is then bound by intrinsic factor, a glycoprotein secreted by parietal cells of the stomach, and the vitamin B12 or intrinsic factor complex is carried to the small intestine where it attached to the receptor and is absorbed. So following the absorption, the vitamin is transported to the liver in the blood bound to transcobalamin 2. So if the absorption of this vitamin can be disrupted, it leads to different defects. So absorption of vitamin B12 can be disrupted by many effects. Among that effect, the absence of salivary gland proteins, pancreatic protease or intrinsic factors, and the defective binding of the intrinsic factor or B12 complex to the receptor cells in small intestine, absence of part of small intestine or stomach, bacterial overgrowth of small intestine, tapeworm infestation in our body, use of anti-ulcer medication which reduce the acid production by the parietal cells, and the chronic malabsorption syndrome, especially which can be seen in an HIV patient, can be lead to the defect for absorption of this vitamin B12 or cobalamin. So this defect can be treatable actually. So 3 types of therapy are there. One, monthly injection of vitamin B12 to bypass gastrointestinal tract, and B12 nasal gel are there. This nasal absorption does not actually require intrinsic factor, and a weekly injection of vitamin B12 supplement in a mega dose, actually more than 300 times a day, allow absorption by passing passive diffusion. Most B12 deficiencies result from what? The problem of this absorption of the vitamin. So the absorption defect can lead what for different problems and deficiencies of this cobalamin. When you see the storage of vitamin B12, the 50 to 90% of the body's total supply is stored in the liver, and the storage so great that the single monthly injection prevents what a deficiency when there is an absorption problem. And this vitamin B12 is continually secreted into bile. Most of it is reabsorbed by intrahepatic circulation. Therefore, little vitamin B12 is lost from the body. Just a small amount of escapes reabsorption are there. So the storage of vitamin B12 can be mainly takes place in our liver. When you come to the advantage of the use of this vitamin B12, actually it is very important and crucial for human beings. There are only 2 clinical significant reactions in the body that require vitamin B12 as a cofactor. There one is synthesis of methionine from homocysteine. The methionine cyanidase enzyme require methylcobalamin as a coenzyme, that means, and isomerization of the methyl, which is methylmalonyl-CoA to succinyl-CoA. This methylmalonyl-CoA mutase requires what? 5-dihydroxy cobalamin as a coenzyme. Therefore, it can be important here as a coenzyme, for the synthesis of what? Methionine, as well as isomerization of methylmalonyl-CoA to succinyl-CoA. When you see the conversion of methylmalonyl-CoA to succinyl-CoA during the metabolism of fatty acid with an odd number of carbon atoms, propanoyl-CoA is produced. So this cannot be used in the citric acid cycle, so it is converted to succinyl-CoA, which is an isomer of it, and which oxidized in the citric acid cycle or Kookeef cycle. This pathway consists actually 3 steps. One is propanyl-CoA plus ATP plus carbonate to produce methylmalonyl-CoA. This methylmalonyl-CoA can be converted to L-methylmalonyl-CoA, and methylmalonyl-CoA also converted to what? Succinyl-CoA, simply by using what? Transferase or isomerase enzyme, and then the 5-deoxycobalamin as a coenzyme. So the enzyme which converts methylmalonyl-CoA to succinyl-CoA is what? Methylmalonyl-CoA mutase, which requires vitamin B12 as a coenzyme. So this vitamin also mainly found in our diet, especially meat, poultry, seafood, and the eggs are the source of this vitamin, and especially rich source are organ meats like liver, kidney, and the heart, and the dairy product also actually contain vitamin B12, and the daily recommended amount of this vitamin or vitamin B12 is almost 2.4 milligram per a day for adults. Its deficiency can be associated with different diseases. Among the most common disease associated with vitamin B12 is what? Pernicious anemia, which is what? A megaloblastic anemia caused by genetic problem in the production of intrinsic factor, which means impaired vitamin B12 absorption. There's also a folic acid trap when vitamin B12 is deficient. Essentially, all of the folate becomes trapped as N5-methyl tetrahydrofolate derivative due to loss of what? Functional methionine cyanidase enzyme here. So this trapping prevents the synthesis of other tetrahydrofolate derivative requiring for the purine and the thiamidine nucleotide biosynthesis pathway. So this leads to what? Impaired DNA synthesis, which lead to what? Anemia. So this also are the deficiency of this vitamin B12. So folic acid can be trapped or methionine can be trapped. This one is a folic acid trap mechanism. As you see, methionine can be converted to SNO-C-methionine, and this SNO-C-methionine is also a source of methyl group. We say so it can be converted to SNO-C homocysteine. This homocysteine can be converted to N5-methyl tetrahydrofuran. That can be again converted to what? In the presence of vitamin B12 as a coenzyme, it can be converted to what? Tetrahydrofuran, or in the presence of methionine cyanides plus this vitamin B12 coenzyme can be converted to methionine again. So this one is a vicious cycle that's shown in our mechanism of folic acid trap. Folic acid trap deficiencies of folic acid and the vitamin B12. One is methylmalonic aciduria. This condition helps to distinguish a B12 deficiency from folate deficiency, because the enzyme methylmalonyl CoA mutase require B12 but not folate as a cofactor in the metabolism of what methylmalonyl CoA to succinyl CoA, and the blood level of methylmalonyl CoA are elevated, followed by the cleavage of CoA group and the elimination of free methylmalonic acid in urine. So this can be a congenital or acquired because of vitamin B12 deficiency. So this also scenario, and the main symptom of this homocystinuria, which is deficiency of folic acid actually and the vitamin B12, and this methionine cyanides require both the vitamin B12 and the folate, and this is actually where the folate trap occurs, and the N5-methyl tetrahydrofuran cannot return to the folate pool without donating its methyl group to vitamin B12. And the main symptoms of the disease associated with homocystinuria is excretion of homocysteine in the form of urine, mental retardation, dislocation of the crystalline lenses of the eyes, sparse blonde hair, and cardiovascular as well as skeletal deformities are what the symptom of homocystinuria actually. The same is true in infants breastfed by vegetarian or vegan mothers can develop B12 deficiency. Anemia is a long-term neurological problem, and the problem may originate in pregnancy if mother is deficient of this vitamin B12. And the adult vegetarian can become a deficient again, can eat eggs, take supplements, and so also. So malabsorption syndrome also there, like disease of small intestine, Crohn's disease, HIV positive patient. Older people may have a trouble absorbing B12 as H cell in stomach is reduced. Neurological problem and elevated blood homocysteine can be again a deficiency associated with this vitamin B12. So the last vitamin among water-soluble vitamin is vitamin C, which is known as ascorbic acid, and this ascorbic acid is derived from glucose via uronic acid pathway. The enzyme L-glucolactone oxidase, which is a flavoprotein, is what responsible for conversion of glucolactone to ascorbic acid. So it is absent in primate, making ascorbic acid required in diet, and vitamins include ascorbic acid and its oxidized form like dihydroascorbic acid, and both are in our food we eat. And this ascorbic acid have it is two forms actually. In as ionized form it becomes what ascorbate, and in its oxidized form it becomes what dihydroascorbic acid. So the ascorbate is oxidized form of ascorbic acid, and dihydroascorbic acid is what the oxidized form of what ascorbic acid. So when it comes to the absorption, storage, and the excretion of this vitamin C, actually this vitamin C absorption occurs in small intestine, and active transport as well as facilitated transport diffusion can be used for what transportation purpose. So efficiency of absorption mechanism increase as intake of vitamin C also increase, and the excretion by the kidney increase with dietary intake. The vitamin C in the body exists mostly in the reduced form, which is called in ascorbate forms, and the high concentration of vitamin C are found in the pituitary, the adrenal gland, white blood cells, and the eyes as well as brain. So the lowest concentration actually found in the blood and the urine, and the amount of vitamin C in the blood and the saliva is very low. So the active form of vitamin C is ascorbic acid, and the main function of ascorbic acid is reducing agent in a number of different reaction. For example, ascorbic acid can reduce iron plus 3 to iron plus 2, and the copper plus 2 to copper plus 1. And not only this, it can also act as a potent antioxidant, protecting against damage by reactive oxygen species, not only oxygen species, reactive nitrogen species, and different free radicals that can be generated in our body. Therefore, vitamin C has the potential to reduce cytochrome A and C of the respiratory chain as well as molecular oxygen. And some vitamin C action are associated with coenzyme which is not associated with other enzymes that it means. So this vitamin C acts in different way to the coenzyme of B group. This can modify a compound, whereas vitamin C may act on the metal in an enzyme. The main function of this vitamin C can be collagen synthesis, antioxidant activity, iron absorption, immune function, and the synthesis of other compounds. So this one is the major function of vitamin C, and but it is very important in the antioxidant activities is vitamin C. So vitamin C in the collagen synthesis, the most important reaction requiring ascorbic acid as a cofactor is what? The hydroxylation of proline and the lysine residue in collagen. So collagen, a fibrous protein actually, which is mainly found in connective tissue, and the enzyme involved in hydroxylation use iron. The iron is converted from iron to iron plus 3. So the iron needs to be recycled back to its original state, so vitamin C reduces it. The vitamin is acting as an antioxidant in this phenomena. So vitamin C is required for the maintenance of normal connective tissue as well as for wound healing, since synthesis of connective tissue is the first event in wound tissue remodeling. And the vitamin C also is necessary for bone remodeling due to the presence of collagen in the organic matrix of the bones. So this vitamin C is very important for human beings. So antioxidant, for example, in vitro vitamin C can be an antioxidant by donating an electron. So how the mechanism of action of antioxidant of vitamin C mean that here this vitamin C have the ability to snatch or removed off unpaired electron. In other language, we say that the ability of scavenging free radical are there. So this vitamin C have the ability to scavenge free radicals. This free radical is highly reactive, which can be lead for different disease in our body, especially different proliferation of cancerous cell can also be associated with this free radical. Therefore, this free radical can be snatched by some antioxidant like vitamin C. So vitamin C high concentration in eyes protect against photolytically generated free radical, and the vitamin C in white blood cells protection against free radical produced during immune function. So it can also be used for iron absorption. So vitamin C in meals facilitates the intestinal absorption of non-heme iron or iron that is not in hemoglobin due to the conversion of iron in gastrointestinal tract to iron 2+. So vitamin C can also be counteract the action of another food component that inhibit iron absorption. When you come to the immune function, white blood cells contains a high concentration of vitamin C, may provide a protection against oxidative damage associated with cellular respiration, and these free radicals generated during phagocytosis and neutrophil activation, which are intended to kill bacteria or damage tissue, and can also kill the body's own immune system. So antioxidant property of vitamin C counteract this. So it also used for synthesis of different compounds. For example, several other metabolic reaction requires vitamin C as a cofactor. For example, catabolism of tyrosine, synthesis of epinephrine from tyrosine, synthesis of serotonin from tryptophan, synthesis of bile acid from cholesterol, synthesis of corticosteroids, carnitine biosynthesis, which is carnitine is actually the transport compound that move fatty acid from cytoplasm to the mitochondria for energy production purpose. So these all are the synthesis of other compound using what? Vitamin C. So vitamin C in diets, the good source of are actually citrus fruit, potatoes, and the green vegetables. And this vitamin C is very unstable when it contact with heat, iron, copper, and oxygen. And the daily recommended daily amount is almost 90 per day for adults men and 75 milligram per day for adult women. Actually, smokers can require more as smoking causes oxidative stress. The deficiency of vitamin C is well known, especially in infants. This deficiency can lead to what? Scurvy, due to the role of vitamin C in the post-translational modification of genes. So, scurvy is characterized by easy bruised skin, muscle fatigue, soft swollen gums, decreased wound healing, and hemorrhaging, osteoporosis, and anemia. So, psychological problem is also in advanced scurvy. And the vitamin C is readily absorbed, and so as the primary causes of vitamin C deficiency is a poor diet and or an increased requirement. So this vitamin C can be easily obtained in different food source in our environment. So taking this vitamin C is very, very crucial for our health. Yes. So this one is the overall concepts of vitamins, which is especially water soluble vitamins. We can proceed for the next tutorial video on the lipid soluble vitamin, especially ADECA, which is very important for for we human beings. So for next period I will prepare for you the part 3, which is focusing on lipid soluble vitamin or ADECA. So thank you for watching, and as I said before also, please try to subscribe and comment on the comment section of our channel so as to improve what we prepare for you guys. So you have to watch attentively, and you have to follow, you have to listen, you have to take this tutorial video as advantage for you, because it is actually what the revision or the repetition for you, so you have to use it properly. Thank you very much.